Welcome. In the previous video, we studied about outbreeding devices. In this video, we'll study about pollen pistil interactions. Okay, pollen pistil interaction. Okay, first we'll see the definition. All the events from pollen deposition on the stigma until the pollen tube enters the ovule are all together referred as pollen pistil interactions. Right. Now, a note: pollination does not always transfer right type of pollen onto the stigma. Right, so there will be many wrong type of pollen may also come and settle on the stigma. We'll see here on the stigma, right, I'll show the pollen which settle on the stigma. Right, here I have shown the different pollen with the different sizes, indicating that these are the pollen grains that have come from different sources. Okay, right, say suppose this pollen is from same flower, right, and this pollen is from another flower, on another flower from different plant but same species, right, and say this is pollen from different species or genus, right. So like this there will be so many type of pollen grains from different sources will come and settle on the stigma here. And the pistil has the ability to recognize the right type of pollen, right. So now here if the plant is undergoing self-pollination, maybe in that case the pollen from the same flower will be of a right type. So in this case I am considering xenogamy, and based on that the pollen from another flower from different plant of same species, I'll consider it as right type. So the right type, which I call it compatible. Right. So now we understood that the pollination always doesn't guarantee the right type of pollen transfer onto the stigma, right. So that is for this statement we have seen a explanation that pollen grains can come from various sources. Only the right type of pollen will be allowed to undergo germination and produce a pollen tube. Right now, this pollen pistil interaction is a dynamic process. Okay, right? And this dynamic process involves pollen recognition. After recognizing the pollen, it will have two consequences. Okay, one, the pollen may get accepted, so that is acceptance of pollen may take place, or rejection of pollen may take place. Okay, right? And this acceptance of pollen will take place, okay, if pollen is of right type. So that is what I'll consider the pollen is compatible, right? And here the rejection may take place, right, if pollen is of wrong type. Okay, and this I call it incompatible. Okay, so the right pollen I call it compatible, and the wrong type of pollen I call it incompatible. So if the pollen is accepted, then it results in post pollination events. It leads to the post pollination events that leads to fertilization finally. Okay, then so that's what the acceptance of pollen, so it will promote the post pollination events that leads to fertilization. Now if it gets rejected, if the pollen is rejected, then in this case the pollen germination on stigma or the pollen tube growth in the style is stopped. So that's what it rejects. The rejection of the pollen will result in the pollen germination on the stigma will not be allowed. If the pollen tube already has formed, then the growth of the pollen tube will not be allowed in case of or in the style. So this is the pollen pistil interaction, that what I call it a dynamic process, and this will involve pollen recognition. After the pollen recognition, that is followed by the acceptance of the pollen. If it is the case, then it will promote post pollination events that will lead to fertilization. So this will happen if the pollen is of right type, that is compatible, and the pollen may get rejected. So it gets rejected at the time of pollen germination on the stigma or the pollen tube growth in the style. So if it happens if the pollen is of wrong type, that is incompatible. Right now, this acceptance or rejection, it depends upon the interaction between the pollen and the pistil. So let's see, the acceptance or rejection of pollen is a result of continuous dialogue between, so it will be a continuous dialogue between pollen grain and the pistil. will be between pollen grain and the pistil. Okay, and this, okay, the dialogue, the continuous dialogue is actually mediated by the continuous dialogue is mediated by, okay, right, some chemical components. So some chemical components are involved, okay, in finding out whether it is of right type of pollen or of okay, wrong type of, okay, pollen here, right. Now this also has, I mean, it also has, okay, some genetic mechanisms. Okay, so these genetic mechanisms also help in recognizing the right pollen. Okay, that is a compatible pollen here, right? And we have come to know that, right, it is all controlled by a single gene called S gene. Okay, so this S gene has several alleles, okay, and identifying those alleles will result in acceptance of the pollen or rejection of the pollen. So what actually all these alleles, okay, and genes and so will slowly come to know in our coming classes. Okay, right. So that's what this genetic mechanisms are actually explained by a hypothesis called opposition S allele concept. Okay, by a hypothesis called opposition S allele concept, and this was proposed by East and a person called Mangelsdorf in 1925. So that's how the pollen is recognized by the pistil. It all depends upon some chemical components, and even there are some genetic mechanisms that help to find out whether the pollen is of right type or it is of wrong type, right. Now this pollen pistil interaction has gained very much importance in plant breeding programs. Okay, so knowledge obtained in pollen pistil interactions. Okay, so the knowledge obtained in the pollen pistil interactions, right, they are actually used by plant breeders. They are used by plant breeders, okay, in improvement of crops or development of superior varieties, or that is hybrids. So in making superior varieties or hybrids, or improvement to the crops, so the knowledge, so how this pollen is being recognized by the pistil, and so all those mechanisms, these plant breeders have come to know, and they are using in crop improvement program, right. And they now as they have gained a lot of knowledge on that, even the pollen from different genera or from different species are made okay, to undergo germination and then fertilize the ovule and thereby producing, okay, new, okay, hybrid varieties. Okay, that so in such experiments, it is important to use only, okay, the desired pollen, okay, from or desired pollen for pollination. Okay, so here the desired pollen means the pollen from a known plant. So in natural conditions we cannot come to know that from which plant the pollen has come from, okay, but in case of these, okay, experiments, they will clearly know that from where the pollen has come from, because they are the one who will collect that pollen and then they themselves dust it on the stigma, okay, and this all process we call it artificial hybridization. Okay, so this artificial hybridization is mostly is a major approach of crop improvement programs. Okay, so it's a major approach in crop improvement programs. Right, this artificial hybridization will involve, okay, two steps, one called emasculation and another step called bagging. Okay, we'll see these, okay, two steps in detail. So how emasculation is performed, okay, and then bagging is performed. Right, first emasculation, okay, is removal of anthers from flower bud before anther basis. So this mechanism or this process we call it, okay, right, emasculation. So this is allowed to remove, you know, or help in preventing self-pollination, okay, right, the emasculation. So here we'll see that one in a flower. So we have here anthers, okay, and the flower is not yet open, so it is completely, okay, closed, right. So these are petals, right, and these are stamens, and this is a pistil. So this process of emasculation is performed by, okay, a pair of forceps. So they use, okay, forceps, and with the help of forceps they carefully, okay, right, keep the petals aside and then completely cut out these, okay, anthers. So they remove the anthers, okay, right, and leave the flower, okay, right, in this way. So now this flower where anthers have been removed, okay, flower in which anthers removed, we call that flower as emasculated flower, okay, and this emasculated flower should be covered with a bag, okay, so this should be covered with a bag, okay. So here you can see, so this is completely covered with a bag, okay, and this bag usually may be made up of okay, or is of butter paper. Okay, so here we use a butter paper to completely close this, emasculated flower. Okay, the emasculated flower should be bagged. Okay, the reason for bagging is to avoid contamination of stigma with the unwanted pollen, because we are bringing the pollen from a known source. Only that pollen have to be dusted on the stigma. Okay, and